So welcome everyone. It's February 13th, day before Valentine's Day, and we are talking about our love for the water. We are very happy to be joined by the research team from Stanford University, Professor Ali Bain and Sarah Lowry, her doctoral candidate. And this is a study that has been a year long in the making. And we're very lucky to have it be based in our community. We have so many people who care for our waterways and our ocean. And we know that the ocean is connected. So it is part of our kuleana to keep our ocean clean because we know it circulates around the globe. So, and also to protect all of us who enjoy it and recreate in it and preserve it for future generations. So what I would like to do is I'm going to ask Sarah to have her presentation ready, and each of you, if you can speak very clearly into your microphones on your computers. We have a portable speaker here, but there's many people in the room, so the clearer you are, the less fuzzy the volume will be. We'll have you present, and then we'll open it up for questions, and we're going to go back and forth so that people in the room are able to ask questions, as well as people who are online. Some people have asked, if you're wondering, this meeting is being recorded. What I usually do is a day or two after the meeting is complete and everything is uploaded to the cloud, I go ahead and send out the link to the recording, which you are welcome to share. Please do not edit it or alter it in any way. We want to preserve the integrity of the data and the results and everything that will be shared with us in this space. And so in the interest of transparency, we ask that you just don't change it or post it, but you're welcome to share it among your networks, friends, anybody who is concerned about our water. And in future meetings, we will be talking about what to do with the results and see if we can play a proactive part as community and as citizens of this place. Okay, so with that, I'm going to mute myself. Everyone who is not presenting, please do mute yourself so that there's not interference. And I'm going to pass it over to Sarah and Ali, and thank you for being here and spending your evening with us.
Thank you so much, Leilani. I will share my screen and we can get started. Okay. Does that look okay for everyone? Great. I don't know if I'll be able to see the chat, so just interrupt me if there's any sort of technical issues or anything. So for those of you that haven't met me, my name is Sarah. Like Leilani was saying, I'm a student at Stanford University. And we have been working on this water quality study at Castles for about a year now. And so we're really excited today to be able to share with you some of the results from this study. So I will dive right in. So I'll introduce Ali's on this call, but it's a little tricky with Zoom. So I'll introduce Ali. Dr. Ali Bain is a professor of civil and environmental engineering here at Stanford University. She's been a professor here for over 20 years, and she's grew up in Kailua. So you can see in her bio here, she paddled for Lani Kai, but she's been at Stanford for the past 20 years, and she's been doing research on Oahu, on Kauai, and on the Big Island throughout her career here. And I am a PhD candidate in her research group at Stanford. I'm in my 4th year of my PhD, and my research focuses on the human health implications of pathogens and indicators in water systems. So I study things like drinking water, wastewater, stormwater, and in this particular project, we're looking at recreational and surface water. I'd also like to introduce the rest of the research team that's been super critical. So first, we've been working with Dr. Oren Shanks, who is a senior scientist at the United States Environmental Protection Agency in their Office of Research and Development. He and his group specialize in the development and use of genetic-based tools for water quality testing. And he's been doing this for a very long time and is an expert in the field. So it's been really great to be able to work with him on this project. In his lab is Adam Diedrich, who is a biologist that helped us run all of the samples for this project, while we were working in Ohio. And then also I'd like to introduce Ella, who may be in the room or on the call. And Ella is a local high school student who lives near Castles. She's been really interested in its water quality, and she's been a really important part of the team, helping us collect samples and all sorts of measurements over there last year when we were collecting samples. So with that, I will refresh the project just in case this is anyone's first exposure to it. So we're studying fecal pollution in the Castles Kawainui area, like Levani was saying. And we have done this by measuring two different categories of things. The first is fecal indicator bacteria, or FIB. These are bacteria that indicate the presence of feces, but they're not harmful to humans. And a lot of you may be familiar with this because this is what the Department of Health and the state measures to post, beach advisories and warnings and such. We also measured in this study microbial source tracking, or MST markers. So like the name indicates, source tracking tracks the source of the feces. So it's a lot more specific than what the fecal indicator bacteria can give us. And so the goal with this source tracking is that you can tell where the feces are actually coming from, whether it's human, bird, dog, or other sources. And again, these aren't harmful to humans. They just indicate the presence of other things that may be harmful to humans. So like I mentioned, we processed water samples from the study actually at the US EPA's research laboratory in Ohio. And so I wanted to thank you all for being patient. I know this has been a long time coming and a long process getting this data, but it's really great. The data has to go through an EPA process before being released to share with you all today. But it's great that we do have that extra layer of checking by scientists in the EPA that have worked with this data. And so I know most of you are very intimately familiar with the potential sources and potential issues of Kailua Bay's water quality, but I'll briefly overview them. So there are a couple of things in this map that I have marked. The yellow stars are the two wastewater treatment plants, the Marine Corps base and the regional plant. And then the blue star is for the outfall on the Makapu Peninsula. The little red dots are the cesspools in the community. And then the green star is the Kawainui Marsh, which we're also interested in. And then the last thing on this map is the purple triangles, and that's where the HDOH is regularly monitoring fecal indicator bacteria, or enterococci, either weekly or biweekly. And so I'll give a little bit more detail on each of these sources and why we're potentially interested in them for this study. The Marine Corps base has been fined a lot of money for exceeding enterococci counts. The regional wastewater treatment plant, I know, was in the news, I think 2 weeks ago now for more exceedances and spilling. So they've obviously struggled with frequent enterococci exceedances. This says in 2023, but we know 2024 and now in 2025, this is still an issue. And both of these treatment plants are discharging off the Makapu Peninsula about 2 miles out and 100 feet deep. We also have cesspools in the area, and in case you're unfamiliar with cesspools, they are pits that do not treat sewage, just holes in the ground, and they pose a potential significant environmental risk. Especially in Kailua, most of them are located really close to the ocean or close to groundwater, and so they could be a source of human feces and therefore a source of pathogens into the environment. Currently, there are about 90,000 cesspools in Hawaii. I know the law was introduced to try to convert them all by 2050, but as of right now, they are around and potentially contributing some health risk. And so as a reminder, this is where the cesspools are located in the community. And so this helped us inform, where we were focusing our study and this project, because they are heavily concentrated in the north of the bay. And this is from the Hawaii Cesspool Prioritization Tool. So if you're interested in learning more about the presence of cesspools, I would encourage you to check out. It's a free online tool that's super valuable resource. And then finally, the Kawainui Marsh and Canal. So the marsh has obviously a lot of water birds, so it's a really important habitat for birds. And we know from previous research that water birds can be really important contributors to fecal indicator bacteria concentrations. And so we thought it was important that we investigated, that particular area as a part of our study. Our group also has done some research in this area. Back in 2011, we measured the stream and we found pathogens including Salmonella, Campylobacter, Staph, adenovirus, and norovirus. While it was now maybe 15 years ago, I think it's still important that we have some contextualization of what we've discovered previously going on in this area. And so that brings us to the main question, the research question that this project is trying to answer, which is, are fecal indicator bacteria at castles from human fecal contamination? And so, like I was mentioning, HDOH and people all over the world use fecal indicator bacteria to indicate feces, but they can come from a lot of different sources. They're found naturally growing in the sand and grass sometimes. They can come from birds. They can come from humans. They can also come from dogs, chickens, cows, etc. But we're really interested in what fraction of this is coming from human fecal contamination, because there's evidence that human fecal contamination has the highest health risk for us. And so the way that we can address this is by using this technique called microbial source tracking. And so microbial source tracking is a lot more specific than fecal indicator bacteria, and it can actually tell us where specifically the feces is coming from. So for example, you can have a bird marker that's specific only to birds and a human marker only specific to humans, as shown in this little diagram. And so that's what we're using in this project. So the formal objectives for this project are, first, to conduct a monitoring campaign to measure fecal indicator bacteria levels in this area in Castle's Kawainui. And then the second is to, alongside that, use microbial source tracking at the same area. And so I'm really happy to report that this project research, the lab side is complete. So I shared with you all last May the results of the fecal indicator bacteria monitoring campaign that we conducted, and I will bring those results back in today to remind everyone what they looked like. And as of December, so just like a month ago now, we have finished the microbial source tracking work. That was all done in Ohio at the EPA. So I'll briefly overview the markers we selected for use in this study. The names are below the pictures. So we have 3 human markers. They're called HF183, crassphage, and tomato virus, and I don't expect you to remember them. We also have a bird marker called GFD and a dog marker called DG3. And you may be wondering why we picked 3 human fecal markers as opposed to just the other markers have 1. That's because, first of all, we're most interested in human fecal waste. And second of all, we selected these targets because if you look at the chart on the right of the slide, they each represent a different type of marker. So one is a bacteria, one is viral RNA, one is viral DNA. And as an example of what these, random names actually are, HF183 is found in your gut, in the bacteria, the commensal bacteria of your gut. So it's in everyone and we excrete it. It doesn't bother anyone. Crassphage is a bacteriophage, so it eats bacteria. It's a virus, and that infects your own gut bacteria. So it's another thing that you don't really notice, but it's specific to humans. And then the last thing, which is a new marker, it's called tomato brown rugose fruit virus. And this is a virus that infects tomatoes and it gets into our gut through our diet. So if we eat anything with tomatoes, whether it's fresh tomatoes or processed tomatoes or peppers, it can get into our gut and we excrete it, and then it can be associated with human fecal waste. And none of these things are harmful to humans. They just indicate the presence of human fecal waste. And we care about these different forms of fecal markers because there are different forms of pathogens. So for example, Salmonella, which you may have heard of, is a bacteria. Norovirus is an RNA virus. And so we've picked each of these markers to try to capture the pathogen variation that we see in the environment, because different types of pathogens might survive better in different environmental conditions. For example, a bacteria may survive better than a virus or vice versa. And if we only have one marker, we run the chance of, missing that relationship. So we chose to include all 3 markers in that study. And I'm happy to talk more about this at the end if anyone has any questions. So for an overview of our sampling, we've selected 3 study sites, the first being at Kaha Park at Kawainui Marsh, the second being near the cesspools off of Kailua Place in the canal or the stream, and then the third being at Castles, where HGOH is already regularly sampling. We conducted 2 sampling campaigns. The first was a biweekly campaign done by Ella from January to April of last year, where she measured enterococcus. And then the second was the daily campaign that we conducted in April and May, where we measured both enterococcus and microbial source tracking markers. Along with all of the samples, we collected some other important data like salinity, water temperature, turbidity, and rainfall to help inform, what these numbers actually mean. And the way that we collected samples is we took large bags and we scooped 1 liter of water for MST. And you can see what that looks like here in the picture on the far left. So we collected the samples in what are called Whirlpack bags, basically just big old plastic bags. And so you can see what each of the samples looks like. You can actually see the difference in each of the sites. The A site is the marsh, and then going down to the canal, and then the ocean is very clear. And so we collected those samples, we put them on ice, and then we split the sample into 2. And with the first part of the sample, we ran IDEX interalert assays, and so that's to detect fecal indicator bacteria. And then with the rest of the sample, the majority of the sample, we filtered it, and we captured all of the genetic material on this tiny filter. And then we shipped those filters on ice from Kailua to the EPA lab in in Ohio, where they have a lot of great molecular tools that we could use in their laboratory there. So in case you haven't seen what a test for enterococcus looks like, this is called the IDEX test. And when it fluoresces, it means that that cell is positive for enterococcus. So this is a sample that we took in the sampling campaign that had quite a lot. And as a reminder, enterococcus are not harmful, but they're indicators of the presence of harmful pathogens. And for the interpretation of this data, when I show you all this, just a reminder that the recreational limit set by the state is 130 MPN per 100 mil. So I'll show these results in the context of exceeding that threshold. And then for the microbial source tracking testing, like I've mentioned, this was all done at the EPA lab, and we extracted the genetic material from the filters, and then we ran all of the MST assays using these molecular methods. It's called polymerase chain reaction, where we're looking for a very specific sequence in the water. And if it matches perfectly, then we can detect it. And so the assays that we ran, or the sequences we were looking for in the table here, you've already seen them before. They're the 3 human markers, the dog marker, and the bird marker, along with 2 controls that we added to make sure that our laboratory tests were actually working correctly. So I'll remind everyone first of our biweekly monitoring results that were all done by Ella. So Ella took 36 water samples, 18 from Kailua Place on the canal and the stream, and 18 from Castles. And she measured enterococcus, salinity, temperature, and turbidity. And in these results from January to April, she found that 50% of the samples exceeded the state threshold of 130 MPN per 100 mil in the canal site. And the median concentration was around 83 MPN per 100 mil. And then it was a bit of a different story at the Castles site, where only 1 sample exceeded that threshold. But notably, that sample was at 460 MPN per 100 mil, so quite a high sample. And we can take a look at what each of those samples actually looked like. I'll show you in a bar chart. So this is what the chart's going to look like. On the x-axis is the date that the sample was taken. And on the y-axis is the enterococcus concentration that's measured by the IDEX test that I showed a picture of. And then the red dotted line is the HGOH threshold. So any bar that crosses that line means that it's exceeding that threshold. So this is from the Kailua Place site in the canal. You can see one really notable event at a really high concentration at the end of April, that really large bar chart to the very right. That followed a huge rain event in April, which is interesting. But you can see, we're pretty consistently detecting enterococcus at this site. And like I said, about half of those samples are in exceedance of the threshold. And it looks quite different when we look at the Castle Beach site, where we have that one really high random detection in early February. But other than that, we're not detecting enterococcus very consistently over this sampling period. And so we took this data And we implemented our daily monitoring campaign in April and May, where we sampled twice a day, every day, in the morning and in the evening for 2 weeks at 3 sites, the 3 sites that I talked about. So we just added the marsh site in because we wanted to get a better idea of what was going on with the birds there potentially. So all in all, we have 78 water samples that we filtered for MST analysis. And we also tested all of those 78 samples for enterococcus. For enterococcus, we found that there was an association between the enterococcus concentration and 24-hour rainfall, which is something that's been observed before. So this is good to see that it's consistent with what other people see. And we also found that there was a significant association between enterococcus and the tidal stage. So when the water is, ebbing out of the stream versus flooding up, we saw a difference in enterococcus concentration. And so I'll show you what those look like. The graph is the same format as the other, but it's all 78 samples on one graph. The colors correspond to the location. So first we have the marsh site shown in green. Second, the Kailua Place Canal site shown in blue. And third, the Castle Beach site shown in red. And so you can see a pretty clear trend just from looking at this with a lot of the really high concentrations at the marsh, and almost every sample is above that H2OH threshold. The canal is, definitively lower than the marsh. We only observed one exceedance in this kind of narrow 2-week period, noting that we didn't capture any of the big rainfall events while we were sampling, but we did capture some smaller rainfall. And then we very infrequently detected any enterococcus at the Castle Beach site shown in red. And so I've shown all of this before, but I wanted to refresh all of you of, what we had found. And now I'm excited to share the source tracking results, which I think should be new to everyone. So as a reminder, we have marker detection results from the 78 water samples that I just showed you in that enterococcus chart. We tested for human, bird, and dog markers. And the way that the data is reported is the percentage of samples that were positive for the marker. So if you see a 40 on the bar chart, that means 40% of the samples that we collected were positive for that marker. Hopefully that makes sense. So this is what the graph is going to look like. On the x-axis, we have each of the markers, and I don't expect you to know the names. So the little indicator below will remind you what that indicator means. So you have the 3 human markers, the dog marker, and then the bird marker on the end. And then the y-axis, like I said, is percentage of detection. So that ranges from 0 to 100%. So now I will show you the big results. So this is all 78 samples that we tested and all 3 sites. The first thing I want to point out is that we did not detect any of the first 2 human markers, which are called HF183 and crassphage. We had no samples that were positive. But interestingly, for the tomato virus marker, which is a viral RNA marker, we had 40% of the samples across the entire sampling campaign test positive for that marker, indicating the presence of human feces. For the dog marker, we had about 10% of samples test positive. And then for the bird marker, which I think is the least surprising result, most of the samples tested positive for bird feces. A little over 75% of them tested positive for bird feces. And I think the natural next question is, what did this look like at each of the sites? And so the next slide I'll show you is broken down by location. So we have 3 charts corresponding to each location that are labeled here. There are 26 samples per location, 78 total. So I'll walk you through each of these charts now. First, the marsh site. We have for the tomato virus, about 1/3 of the samples were positive for tomato virus at the marsh. Almost 25% of the samples at the marsh were positive for the dog marker, and every single sample at the marsh was positive for the bird marker, which is not surprising because we know that there are a lot of birds in the area. But it was interesting how high the detection of dog marker was. So dog waste is certainly getting into the water here, and we were able to detect it. So that's good to know. The second graph, which is labeled B, is the Kailua Place Canal site that's kind of adjacent to the cesspools. You'll see that the tomato virus detection rate is a little higher than what it was in the marsh. It's at almost 60% compared to around 33. So we saw yeah, almost 2/3 or over half of our samples were positive for the human marker at this site. And then for the bird marker, again, we saw all of those samples were positive for the bird marker. We did not detect any dog at this location. And then finally, at the Castle's Ocean site, we saw a bit lower detection rate of tomato virus compared to the other two sites, but we still detected it in almost 30% of samples here. We had just a little bit of dog that tested positive. And then we also had a much lower detection of bird indicator at this site at just under 30%. So that's what those results look like broken down by location. And so I want to wrap up with data interpretation, which is where we are at now in the process. So the first thing I wanted to flag was that most of the samples we tested in the lab were below the limit of quantification, which means that they were really, really low numbers that we couldn't actually assign a number to, which is why we just assigned them a positive or a negative, a detect or a non-detect. So that's why you don't see, concentration numbers in this data. And this is in line with previous microbial source tracking marker studies. For example, the bird concentrations are very similar to what other studies have found in the Gulf of Mexico, for example. We detected the bird marker, like you saw, in all of the samples in the marsh and the canal, which is maybe not too surprising for people who have been there. We did detect the dog marker sporadically in the marsh and the ocean. So there is dog waste in the area that is certainly affecting the water quality in this area, about 10% total of samples. And then I think most interestingly, we detected the tomato virus human marker in about 40% of our samples. And what really is interesting is that we only detected the tomato virus marker. We didn't find the other two. And so we've been thinking a lot about why this could have happened. And we have a couple of ideas, but we're not sure. We're not positive yet because research we're not certain. So the first thing to note is that we did use a different machine to detect tomato virus than the other two. We used a much newer machine because tomato virus is a newer marker. And so that may have increased its detection rate compared to the other two. There also may just be a difference in the persistence and the survival of the tomato virus in the environment because it is a viral RNA and the others are a bacteria and a viral DNA. So there may be some, difference in behavior that we don't really know what's happening, that we're detecting that and we're not detecting the others. And then also, most importantly, this is a really new marker for science. It's never been tested in Hawaii before, and it's not been tested in many places at all. And so it definitely requires further research and investigation. And so we're thinking of that as we move forward. And so I'll wrap up by saying the next steps for our research team are to complete the statistical analysis of the data. And so one of the big parts of this is looking to see if there's a relationship between the markers we detected and any environmental factors. For example, are we seeing a marker more commonly after it rains or not? That would be an interesting relationship to see. And then the final step of this project is going to be to publish the data and the analysis in a scientific paper. And I will follow up with you all and share that final data and final paper. So I'm now happy to take any questions that you have. And thank you so much for your time.
Thank you, Sarah and Allie. Let's get them getting applause from everyone in the room. Thank you for that. That was extensive. And I know that I have a lot of ideas in my mind, which, we probably will have to speak at length with community. Just wondering about, what are some of the causes of the human detection over at Kaha Park? Does it have anything to do with, houseless populations? So what I would like to do is open it up for questions. I don't know if you want to keep your screen on so that you can go back and refer to the data. That might be a good idea. But I have one question that was passed to me, and then Gary or folks in the online, if you have a question, please raise your virtual hand if possible. And I'm going to ask Gary to help me call on people online because I'll be calling on people in the room. So the one question that you're being asked is, why not sucralose, used as a marker? Why not sucralose used as a marker?
Yeah, that's we definitely could have used sucralose as a marker. Our lab at Stanford, we're primarily a microbiology lab, and so we used all microbiology targets. But I know Ali has previously done work, I think, with caffeine as a marker, and so those are absolutely valid things to do. It's just not something we did in this study.
Okay, I'm going to take one more question in the room, and then Gary, you can ask for someone online. Steve, if you would speak up so that we can hear you in the speaker.
What if any, illnesses or infections are known to be caused by the tomato virus?
Yeah, that's a great question. So the tomato virus poses no danger to humans, but it is really bad for tomato crops. And so the USDA actually has like banned it, and they inspect fruits and seeds for it that are imported into the US because it has the potential to like decimate crops. But as far as human risk, there's no human risk. It's just something that's become very prevalent in a lot of countries around the world that produce tomatoes, and we eat them. And so it's consistently shed in human feces.
Maybe I can
Sarah,
add
just
a
a
little
point
bit
of
to that.
clarification. Oh, go ahead, Ali.
Oh, I was just going to add to that a little bit, Sarah. The tomato virus is in super high concentrations in sewage and in stool from humans. And it's because the tomato sauce and the other tomato products that we eat in our homes, has the RNA from the virus in it. It doesn't have infectious virus. So it is not, we're not measuring it because we're looking for the tomato virus. It's an indicator of human feces. And human feces can also contain things like norovirus or other pathogens that can make you sick. And so this is like, it's in much higher concentrations than pathogens. So we measure it as a marker for wastewater and sewage and stool in the environment.
Go ahead.
So what's the most likely cause of staph infections that people get in Kailua Bay? What's the source?
That's a great question. I don't think that I have a good answer for you now, unfortunately. I think this is kind of the first step, like an exploratory study, because we weren't even sure if we were going to find evidence of human fecal contamination in this area. And so I think an important next step for a future research area would be to try and maybe explore that a little more. But unfortunately, I don't have a good answer for that now.
I have one question for Ali. The wastewater dashboard that you created for other projects, would that be helpful to look at the data coming from that in the context of some of these results?
That's a good question, Levani. I think that, I mean, we have samples archived from the Kailua wastewater treatment plant, the sewage. And we could go and measure the tomato virus in there and other pathogens. So that might be useful. I mean, I put the link to the dashboard in the chat for people who are curious, but the data on that dashboard might not be useful, but the samples might be. And so I think Sarah and I have chatted about that a little bit. I also wanted to say that the question about the staff, just to be clear, our research question was not, what is the source of staph? Our research question was, is there human fecal contamination or other animal contamination in Kawainui and at Castle Beach? So we didn't set out to figure out the answer to the question about the staph aureus.
Gary, is there someone online who has a question? Or you can ask.
I sent you one text already that somebody wrote. It's about the tide. What was the tidal conditions during the test period?
That's a great question. So we designed the study so that we'd have equal ebb and flood tidal conditions. So we have like a paired data set. We have about half of our samples that are ebb and half that are flow. And I also recorded, the actual tidal levels from the station over in the next bay. And I didn't share those results because I'm not quite done analyzing if tide has an impact on marker detection, but that is something that we're looking into.
We have another question. would the eDNA be the next step to determine bird species and their source? Having a hard time reading these things.
Thank you. Yeah, so I do not personally have experience with eDNA, but I know our lab does do a lot with eDNA. I think in terms of health risk, I think the next step for me personally would be focusing more on the human side and less on the bird marker. But eDNA is a really valuable tool in assessing, you know, what actually is going on in the water. Maybe Ali, I don't know if you have any more thoughts on that.
Another-
About the eDNA, and that's related to Lisa's question about like doing sequencing of the samples. And yeah, I mean, we have nucleic acids like from all these samples, so we could measure different things in them if we wanted to. We're mostly interested in viruses, which are RNA and don't have like a barcode that you can just, you know, amplify all the viruses, but we could do something creative with that. And then for the eDNA question, I think if you want to know what kind of birds are contributing feces, I think you could just go to the marsh and look at the birds and like identify them, because there's lots of birds. But yeah.
Okay, so I have a two-part question, and then Gary, we'll go back to you. Would any of these 5 markers detect other potential sources such as pigs, cat, and mongoose? That's the first part. And then the second part is understand the human viral rate would be higher in cesspools, 60%, but any explanation on why it is so high, 30% in the marsh? And then we'll go to Carmela.
Yeah, so to answer the first question, I think about like how specific these markers are to the targets. So these markers have all been, you know, pretty extensively evaluated to check for cross-reactivity, to check that like mongooses don't also have this marker, pigs don't have this marker, cows don't have this marker. And so we do have pretty good evidence. I think we have the least evidence for tomato virus since it's the newest, but we still do we have tested to make sure that like cats don't have it and dogs don't have it. And so there was a lot of vetting that went into these markers to make sure that they're not cross-reactive with other species. I'm not entirely, I think that there is a pig marker that is in use, but I'm not entirely sure about that. And we did not choose to use it in this study. And then remind me of the second question, sorry.
Yes. So the second question had to do with the human viral rate would be higher near the cesspools, 60%, but any explanation for why it's so high in the marsh?
Yeah, that's a great question. I was also a little surprised by how high it was in the marsh. I think it could be worth looking into seeing like when runoff is happening, where runoff is coming from. It could be worth examining the like septic or not septic, the sewage lines in the area and seeing if there are any sewage lines. I know that the sewage line crosses under Kawainui at some point, but taking a closer look at all of that, but I don't have a satisfying answer right now.
Thank you. Carmela, if you can ask your question.
Great talk, Sarah. I really enjoyed it. I've learned a lot about microbial source tracking from you and Ali's research. And I just wanted to ask a question. I'm curious a little bit, a lot of your data, right, it's all percent detected. Is there another method where you could look at concentration or abundance from these different sources at all, or how you would characterize their contribution overall? Thank you.
Yeah, that's a great question. So I know working with Oren at the EPA, his lab does a ton of microbial source tracking. And so from my more limited understanding, the bird marker specifically is typically found at just consistently low levels. So we're using qPCR and the CT values are just below the lower limit of, or above the lower limit of quantification consistently. And so that's a normal finding. For the tomato virus, we were using dPCR with the QIAcuity machine, and we did have a couple of samples that were above the limit of quantification, but it wasn't enough to make like a complete picture. Like it was like very few. So I think that was a result of just these specific samples. I think larger sample volumes potentially could have helped us reach higher numbers if we were able to filter more than a liter. But that's kind of why we were limited by the percent detection in this case.
Ali, do you just want to chime in? I'm not going to open up the chat because there's all these people, but can you just chime in on what you felt was a misinterpretation?
Yeah, someone thought that we might have said that there is no risk of from exposure to the water from the study, but that's not what we said or meant. We meant that if you're exposed to this tomato virus, the tomato virus isn't going to make you sick. But I'll go out on a limb here and say that if there is, the results suggest there is like human waste in the marsh and bird as well. And so it's probably not a good idea to swim in the canal or next to the marsh. But I think everybody knows that already, but this just confirms that it's probably not like a very good idea. And we don't have risk estimates. It's something we could eventually do. We could model the risk of illness from exposure to the water with more work and stuff, but that's not part of this particular study. Again, our research question was, is there human feces and sewage in the canal and at Castle Beach? And that was how we designed the study.
Thank you. Someone else online who has a question? I
well,
can, oh, go ahead, Gary.
There's a question here. I heard the Kinky Ball campaign close to the marsh. Was this considered as a possible factor that could skew the results or help reduce harmful levels?
Correct me if I'm wrong, Carmela, but I think they were at different locations, right? So this was Kawainui and that's Hamakua. So no, yeah, as far as I'm aware, not potential for interference there.
Next question. What is your assessment of how bad are these conditions on a scale to 1 to 10?
That's a good question. I don't know if I can give you, I feel like I'm saying this a lot. I don't know if I can give you a satisfying answer. I do think that it's a really significant finding that we did detect human marker because previously, we only had this intercoxy data and we weren't really sure where it was coming from. And now we have, a little bit of evidence where we can say, hey, there's definitely bird feces in here, but there's also potentially human feces, which is a problem for human health. So I don't think I want to give a number, but I do think that it's significant in a health importance that we're detecting a human fecal marker.
Next question relates to the homeless people near the park and the possibility of them creating the feces in the water. And this person says they launched their canoe at this point and frequently hear voices coming from the area. I myself have experienced that when we cleaned out the mangrove, that as soon as we got rid of all the homeless people, I was testing the water in that canal because we had 20 employees in the water clearing out the mangrove. And back then, the test results was 1,700 times the allowable amount and 1,600 of human. So animals was the bigger number. And as soon as we cleared out the stream of all the mangrove, those numbers went way, way down and we got rid of the homeless people. But they're all back now again. That might be.
I just want to interject and say I think we need to be really careful about blaming unsheltered people. And there are strategies to provide like sanitation facilities perhaps. And as you guys all know, some of the unsheltered people are suffering from mental and physical illness. So I just want to be careful that we don't start sort of, I think it is a valid point, and thank you, Michelle, for raising that. And I think we have, we've seen some of these folks, so we know about them. But I think there are some solutions, like perhaps providing porta potties. I know there's a bathroom over by the airplane field where some of them can use the bathroom. I don't know if it's locked, but it's definitely a societal challenge. But I don't think we should focus too much more on that.
Thank you. We have a couple of questions here in the room. Go ahead, Michael.
I wonder.
Come to the speaker in case she can't hear you.
I wonder whether you were curious and looked at results of studies at other swimming beaches on the California coast, or maybe I doubt whether you'd find them in the islands, but there must be other studies. And I'm guessing you were curious to see how we stack up. Do you have an idea of how our results look against other prime swimming beaches in similar, like on the coast of California?
Yeah, I'm not sure about the coast of California specifically, but I have been looking at, US-wide studies of these types. I think from what I've seen, and this will be something that I'll, write about more formally, and I'll be happy to share with you guys once I've, conducted maybe a more formal analysis. But for what I've seen, the bird concentrations that we're seeing are very similar to what other places have detected, as are the dog concentrations. They're about on par with what other studies have found. It's a little bit harder to say with the human, because with this tomato virus marker, this is, one of the first uses of it. And so there's not really a precedent for that, not really a good comparison for that, because it is relatively new. But I will look more into what other human markers, what they were detected and how they were detected. And that's something I can write about and follow up with you about.
I was gonna add something, Sarah, since for better or worse, I'm the poop queen and I've been studying dirty beaches for 20 years. So I can say that even in California, at the most like contaminated beaches, that detection of like the HF183 marker is really rare. So it's not surprising that we don't detect it, and we could detect something else. It's usually in pretty low concentration. So I'm trying to think of like a famous beach that's really contaminated, like Avalon Beach on Catalina Island. There were like leaking sewer lines there, and we just like sporadically would detect the HF183 marker over like an entire month. So these results with the older markers are not like that different from other places, and we do expect low concentrations.
So we have another question in the room, and then I have a question from the community perspective. Go ahead, Doug, ask your question.
First of all, I just want to say thank you for making this very clear. It's really helpful to us to understand what's going on. Now that you have this technology that allows you to do such a thorough study of these MST markers, one thing that this potentially reveals is us to consider all the data that's been gathered in the past that has not shown the presence of a human fecal matter to be significant. Perhaps it has always been there, but without the lack of this new technology that really does reveal it, we just haven't found it. But now we are finding it. And the second thing is the person who raised the issue about the unsheltered. Let's keep in mind, there was significant result here from these cesspools, extreme result from the cesspools. I can pretty much say that it's unlikely that any of those homeless people are contributing to the cesspool leakage. So let's keep it in that context.
You want to respond, Ali or Sarah? Then I have a follow-up.
Thank you. I don't think I have a response, but thank you for bringing all of that up. I think it's a great point that, as technology advances and we find these new markers, we may find surprising results that may perhaps have been there all along and we just didn't know. So that's a great point.
Okay, this question also relates to cesspools and tides. Could inflow of tide in the channel, theoretically with high tomato virus marker from cesspools, be accounting for higher readings at Kaha?
That's definitely something that we're considering, is the influence of the tide and, how the water pushing back and forth, especially if there's, a certain level in the cesspool and maybe the water level is reaching that as the tide rises. That's definitely something we're considering. And that's why in our, study design, we kind of split up. We made sure we had both ebb and flood tides represented in our data set, and we will be looking into that. We just aren't quite there yet.
Okay, so I have a follow-up, and then Gary, you're welcome to ask your question. My follow-up is just from the community perspective. Our subcommittee largely is about empowering people to be informed and advocating for things that will help us with water quality issues. So do you feel, based on your results, that it would be worthwhile for us to pursue this cesspool issue? And again, we don't want to just target, but we're saying that it's relevant to us because we have a high concentration of cesspools in an area of our community that are close to the beach. So we're not trying to blame and shame folks who happen to live there. But as a community, do you think we can get behind them and support efforts to faster convert them to sewer systems, city sewer lines, based on some of your results? And do you think that would have a positive impact?
That's a great question. I think maybe the hardest part is that this is, the first study that I'm aware of in this area that's been looking at this. And so we're dealing with, for the canal site, I have 26 samples in April and May. And so I think it's just best to caution that this is exploratory. We didn't capture any huge rain events. We may not have captured perhaps seasonal variation or something. And so while this is really important data, I don't know if we have enough evidence to fully commit to the cesspool, but it's certainly a promising theory. And so I think from my perspective, a little more research may be necessary, just because this is such a small sample size and a small point in time. Yeah, I don't know if you have any thoughts, Ali.
So
Yeah,
future doctoral students, Ali and Carmela, future students are welcome to do studies.
Yeah,
Go ahead, Ali.
if you ask a scientist if they have enough data to say anything, they will always say they need more data. But I think the Sarah is writing a paper right now, and Oren, who is a longtime friend and collaborator at EPA, will be a co-author on it. And I feel the conclusions of that paper are going to say that there's evidence of fecal contamination in Kawainui and at Castle Beach. So armed with that, co-authored, well, are we still recording?
We are recording, but it's a fair point that the sample size was kind of small and you had a very limited time to do this. So that's understood.
Right. But
we understand
I
those
think
constraints.
Sarah has been really clear that we have a collaborator at EPA on this project who adds clout to it. And I think that was one of the exciting things about this project, is we thought that we could bring that to the community and have a really someone at EPA who's engaged in this project. So it kind of has that additional gravity associated with it, right? So, I think that you guys will have some information that you could go to DOH and show them, and we'll be sharing this information as well to DOH, but they were very interested in this study, and we promised we would follow up with them on a Zoom call to talk them through it. So Sarah, that's something that we should probably do in the next 4 weeks or so.
Okay, Gary, go ahead, and maybe we'll see what your question is, and we may take just one more because Ali and Sarah have been kind enough to give us all of their time. So go ahead, Gary.
I have a two-part question. First part is, would it be possible, it doesn't have to be officially in your document that you're going to produce, but would it be possible to also throw the data from the Department of Health at those other test sites into your charts and graphs as an afterthought? Because you didn't do it, so they did it, and so I can't expect you to validate the data, but it would be interesting to see if their test results kind of match yours, or maybe add to yours.
Yeah.
That's my first question. And then the second question is, I had done some research just before the virus hit, and there's a guy that was an engineer with a sewage treatment plant on the mainland, and the plant was very huge, and he got very tired of driving all the way to the other side of the facility. And so he invented like a stainless steel box on a pole with a solar cell, and it tested all the water leaving the sewage treatment plant. And then he had that data uploaded into a web page that he had, and the whole community could see his results, and management could see it at the same time. And I understand that Pearl Harbor has been using that company as a test to see if that results is valid or not. Do you have any thinking about these self-testing units on an ongoing basis?
So for your first question with the HDOH data, that's a great idea. I have been referencing it throughout this project, kind of side by side. I think one tricky thing is they don't sample after it rains, and they only sample every other week at castles. So it's a pretty limited data set. I also was on the website a couple weeks ago, and I think it's undergoing construction. So I don't know if anyone knows about that, but I was having some trouble accessing the archive. But I do have like screenshots of the data from last year, and that's definitely something we can add in like the supplementary of the paper if it ends up being like useful, like it overlaps. So that's a great point. As to your second point, I personally am not familiar with this technology. I know it's notoriously very difficult to measure microbial things with sensors. I don't think there's been a lot of success that I've been aware of, and this project is specifically focused on, pathogens, but I am not familiar, so I can't really speak to that, unfortunately.
Thank you.
Yes. Okay, we have one remaining question from Derek. And I'll make the caveat. I see there's 19 messages in the chat, which I will save. Anybody else who wants a recording or who has follow-up questions, maybe after you leave this meeting, you can email me and I will get it to Sarah. Derek, go ahead and ask your question.
Sure.
Before we adjourn.
Again, mahalo nui loa for your study and for all your work here. A big shout out to you, Ali, from Tom and Kirsten over here in Kailua. And so just the presence or absence is indication enough. No real need for quantification. It's just, is it here or is it not here? And then the likelihood of it actually getting brought in from the sewage treatment plant, I would think is a pretty low likelihood, and that it's more likely due to some other means of leaching into the system rather than the sewage treatment plant. Is that am I barking up the right tree on that?
Yeah, we tried to pick the sample sites, pretty intentionally. So we had the ocean one, and we could compare, that ocean one to up the stream. And so I do think that is in line with what we've observed in the results here.
Yeah. So one thing that, perhaps I can work with you on is at least putting out maybe a shout out for future studies or areas that might need to be investigated. Certainly, my personal opinion is waiting until 2050 for conversions is too long to wait. And that's just the aspect of the cesspools. We have some other anecdotal things that we've learned recently. There's no longer, it used to be Kaha Park, used to be a dog park. The city is not allowing that anymore. So they recently announced at our neighborhood board meeting that you are not to bring your dog, that you will get fined for bringing your dog in that particular area where a lot of people were using it as a dog park. And then we discovered in another part of the community that someone was putting their dog poop bags in the storm drains up on the Kala Hill hillside. So we did a community activity this last weekend where we did some storm drain stenciling, drains to oceans, and education around that. But you know, I do worry. We've had some very big rains lately, and I think it would be awesome to kind of send out a team every time there's a big rain to go out and quantify. That might help us identify. So maybe that's a future study. So, but I just want to thank you both. I know it's late in California. I want to thank you both for being here and for doing this, and Ali for just having the you know, the determination to just do this with your funding that you had, and be willing to work with us and come back and share the results. Certainly, I don't want people to jump to assumptions. I believe in research. I believe in science. And I know that you all have a very high standard. So I just want you to know that I recognize that and appreciate it. And so I'll just leave everyone with the thought that the work remains. We have a lot of work to do. And this is really ground up. So community empowering each other. Stay tuned for the emails. And next month we'll hear the results of the Genki Ball study and the stormwater town hall that Councilmember Kia Aina hopes to have. So thank you very much, everyone. This meeting is adjourned.
Thank you, Leilani.
Thank you, Leilani.
Oh, you're welcome. Pleasure. Thank you for everyone who's here. There's a lot of people in the room that you can't see, but thank you for your presence. And please go home and share this with your friends and neighborhood. Okay. All right. Thank you. And thank you, Gary.
You're welcome.
Okay. You want to say it here?
Yeah, I'll say it there.
One observation.
I was just on Kailua Beach, and it was a very surreal occurrence. It was right after a heavy rain. The guy from the health department was posting trees at Kailua Beach. I walked down. Now that there's a lifeguard stand, there are no Portuguese men of war, but there's a warning for Portuguese men of war. There's a warning for riptides, no riptides. And there are several families with lots and lots of kids in the water that's just been posted. I think we need to get on water safety to have bacterial count warning signs.
Okay.
So can I add one thing to that?
Yes.
Okay. So if I understood them correctly, they sort of gave us a warning that people shouldn't swim in the marsh and shouldn't swim in that canal. One of the problems is that there are some high schools that use that canal for kayak training, because there's kayak competition, single person kayaks, and then also for canoe training. And, you know, that's troublesome. The other observation is that, you know, I would think that the results, if you tested the results over by buses on the other canal, they would be very similar in terms of contamination.
Okay.
Okay.
Oh, yeah, test.
You know,